guys are ready. Oh man, it's on YouTube too.
If you wish.
No, that's fine. I don't, whatever. What could possibly, no, it's fine.
Okay. All right, so let's get started then.
Should I go ahead and share my slides then?
Yes.
Okay.
So welcome everyone to our Cyber Physical Systems seminar today. I'm pleased and honored to present to you Doug Densmore. Professor Densmore is an associate professor in the Department of Electrical and Computer Engineering at Boston University. I obviously have quite some memories, also personal memories, that connect me to Doug, because it was one of the first phases that I can remember also when I arrived at Berkeley. And he was in the corridor of Cody Hall as a postdoc or senior PhD student. And so Doug is the focus of his research is on developing tools for specifying, designing, and for the assembly and testing of synthetic biological systems. And he shows us how actually we can take advantage and leverage concepts from embedded system level design, electronic design automation, such as abstractions, and rigorous ways to transitions between them through synthesis and verification, how actually you can apply this to totally different domains, right, that people would consider unconceivable in a sense. What he calls indeed hardware, software, and wetware co-design, applying this to biological systems, to microfluidics. And we will learn more from him. Just want to add that in addition to the numerous awards that he has received, he's also conjugate his academic career with entrepreneurial career, and he has been founding 2 companies in the realm of synthetic biology. So with all that, thank you, Doug, for being with us. Leave the stage to you.
Okay, thank you, Pierluigi. Thanks for the introduction. I was just talking to Pierluigi about the fact that I first started grad school in 2001. And I was like, oh my goodness, it's been going on 20 years. And I've told a lot of students that are struggling at this time, like what a strange time this is to be trying to go to school with COVID. I started in 2001 in September. So those of you in the US know, right, September 2001 was kind of a crazy time. That was right after 9/11. And I remember there was a lot of uncertainty then too, a different type of uncertainty, but I'm pleased to be here and I'm pleased to be able to talk to you guys. Luigi, I don't know, I have trouble on these Zooms, like trying to take questions and things with chatting. So I assume there'll be some kind of Q&A at the end. Yep. Okay, I'm getting nods. Okay, so I'll just kind of do your best, hold your questions, and we'll just kind of talk about that at the end. I'm going to move quickly because I have lots of slides. But what I want you guys to get from this talk is that my PhD was purely in embedded system design, purely, no biology at all. And then I spent a 3-year postdoc thinking about how we could take techniques from EDA, electronic design automation, and apply them to this new world of synthetic biology. And you'll see how I made that transition. You'll see some of the tools. So when I begin here, what I see here is what you're seeing on the screen is a bee larva, a honeybee, and you're seeing what happens is that in the honeycomb, the bee larvae consume food, they grow, they transform, and they eventually become bees. And when I saw this years ago, I would have said, oh, that's cool, that's a bee. Now when I see it, I see computation, because what you have is a bunch of parallel programs all running at the same time, producing effectively the same result, almost deterministically. Now, every bee is different. Just like your mom told you, you are a special child and you are very different and you are very special. But effectively, as humans, we're all the same, and we all start from a single cell. And that single cell has to make lots of different decisions, but eventually becomes a living computing system. And our brains are a powerful example of biology computing. And so the questions I kind of get into is that biology is another form of information and therefore is another form of technology. And so I've always asked myself, what's the computer science of biology? What does that mean if we said, what's the computing aspect of biology? But programming biology is fundamentally different than computers. It can self-replicate, it can repair, It can change over time. So there are interesting aspects to this. And so really what I'm looking at is how the 21st century computing can expand to inform, influence, and inspire biology. You've heard kind of a biologically inspired computing, where biology is inspired, neural networks inspired, certain different ways we model avionics and things. But think about the other way, where computing could inspire how we design living systems. And that's kind of my approach. So instead of biology inspired computing, it's computer inspired biology. And what's an example of some of these things? These are some examples from colleagues of mine, particularly at MIT, Chris Voigt. You might say, well, what do you want to do? You want to regulate a chemical process where sugar is consumed to make different chemicals. That fermentation process is now like a feedback loop. So you can think of it as a circuit. So you're seeing here that there are this, you might expect in electrical engineering, this feedback loop, and then you're seeing here a genetic circuit, which I'll describe later, that might be able to do that feedback loop. You might think about gene therapies. You'd want to make smart cells that can detect specific inputs, cell types, and maybe make therapies to fight cancer. You might want to make smart bacteria that detect a certain pH and being in the stomach and release a therapeutic when they're in your gut. So think of a smart personalized medicine technique for any number of intestinal disorders. And then of course, smart plants that can sense the amount of water, how old they are, the temperature, and then produce resistance to drought or produce pesticides. So it's not that we try to think of biology as computing to like beat a computer. It's that we think of that this biology needs to make decisions, and biology makes decisions all the time now. What cell becomes a facial cell for me versus an eye cell, or as we watch bacteria differentiate or sense each other and move. They're making decisions. We just don't think of them like that. But if we think of them like logical decision making, we might think about how we engineer that quote unquote digital logic in living cells. And so if you look at a living organism like a cell, you can think of it as maybe having three capacities. It has the ability to sense. It could sense secondary metabolites in the environment. Quorum sensing, that means sensing the presence of other bacteria, maybe their nutritional state. It can take inputs from the environment. You'd want to process that through some sort of internal circuitry to actuate, to make small molecules, maybe to rebalance the gut environment, et cetera. So again, it's got inputs, it's got outputs. Its outputs are typically now proteins, its inputs are proteins or small molecules. And what we need to think about as computer scientists and electrical engineers is how we can think about how we design electrical circuits and work to design genetic circuits that can perform this kind of processing. This field is called synthetic biology. I don't have time to go through the whole primer. I'm just going to pull some images up and talk about them. What you're seeing in the upper side of this slide is that the high idea is DNA becomes RNA becomes a protein. So all that's called the central dogma. All of this talk, and not all of it, but much of the genetic circuit work is how to enable this. So if you see this purple arrow is a gene. Ultimately, we want genes to be expressed or translated, which means that a protein is produced. So this is a stand-in. This is just like when you see an OR gate, that's a stand-in for transistors. When you see this symbol here, it's a stand-in for DNA. And this horizontal arrow is a gene. This curved kind of arrow is a promoter. That's where transcription starts, the process of becoming mRNA. The process of becoming RNA. A terminator is where RNA stops. And then typically they often need, in prokaryotic systems like bacteria, they also need something called a ribosome binding site. Again, it's not important you understand all the biology, but what is important is that DNA has certain rules for how it works, and synthetic biology looks to encapsulate those pieces as parts. Just like we have transistors, resistors, capacitors, and if you don't hook them together correctly, you don't get a functioning circuit. If you don't hook DNA together correctly, you don't get a functioning DNA circuit. And so the people have put these parts into registries. So now you can go and look for these bio bricks where you can assemble composite devices and start to think about making little sensors for environmental toxins, for signals inside of your body potentially, or sensors for light, heat, lots of different applications. And there's a whole industry around synthetic biology. I don't have time for this. This is something you can go to Wikipedia and find, but the general idea for how most of this works, there are different systems. There is yeast, there are mammalian cells, but the classic one is bacteria. The classic one is E. coli. And then what you see on this slide here is that bacteria inside of them, many of them have circular pieces of DNA called plasmids. What's nice about those plasmids is that if they're designed correctly, they will replicate with the cell. So once you introduce one plasmid to bacteria, when it replicates, it will continue. And so on this circular piece of DNA, certain DNA segments have certain properties, not the least of which is you can cut them apart. What you're seeing here is that you can take this circular piece of DNA where my mouse is, you can add an enzyme that cuts it open. A's bind to T's, C's bind to G's. And so what you can do is you can write your own piece of DNA, cut open a circular piece of DNA in an existing bacteria, stick it in. There's a process of hydrogen bonding and ligation that's going to stick it back together. And then you're going to go through a process of putting these changed pieces of DNA back into bacteria. And they're designed in such a way that if the bacteria do not get your design, they either die or change color. And the ones that aren't dead or aren't changed color, you can collect and repeat the process over time, growing up more and more of the bacteria that have your circuit, till eventually that's the overwhelming bacteria in a population. And that's the general idea of cloning. So it builds on this same idea of cloning, where you have a process of introducing DNA into bacteria and then harvesting them or picking them and reprocessing that. All of this is basic molecular biology. It's very common today, but what is different is how we design this red piece of DNA. That's our circuit. In the past, people would do it by hand, and what my group does and what my research is how to do it with computers, how to compile code into these DNA segments we're going to put into bacteria. So an example is a knot gate. So this is a picture of a knot gate. Again, I'll do my best. There's a lot to cover, but what you have to kind of understand is that, again, this is the DNA, this horizontal gene. Pierluigi, can you just nod if you see my cursor moving? Is my cursor moving? Okay, so people can see this cursor. This horizontal arrow is the gene. And what happens is that if you turn on this gene by turning on this black arrow, this protein is made, it floats out and then shuts down another location where you could actually turn something into RNA. So if this gene is on, it shuts off another gene. So if you have a 1, you get a 0. So if you look at this as output, RU is relative expression units. We're measuring the fluorescence. As we increase the input, we get less output because you're getting some amount of protein. And it's not a 1 or a 0 in that way. It's the DNA making a certain amount of protein, and you can, just like voltages are on or off at some kind of abstraction of a voltage, we do the same thing with a fluorescence marker that we'd have in here. And so then a NOR would be the idea that we'd have 2 of these. And if either of these are turned on, then you will shut off. So again, anytime you have a 1, you get a 0. The only way this is not shutting off is if neither of these are present, you have 2 zeros. So you get a NOR gate. Most of the people listening to this seminar will know that NOR gates are functionally complete from a Boolean algebraic standpoint. So if we have NOR gates and NOTs, we can effectively make DNA that implements any arbitrary logic expression. Now, that's theoretical. How we actually get all this DNA to work is the purpose of a lot of this talk. So what we're doing is we make a gate library. And again, the point is not to compute, to beat a computer. It's to have these logical expressions that say, if this signal produced this therapeutic, if this biomarker produced this fluorescence that's sensing something in the environment. So what we do is we have a bunch of sensors that can sense different small molecules. And then we have a gate library. They all have these different transfer functions, which relate to the function I just showed of their input-output relationship. And you have this truth table of a circuit you want. And if I told you, okay, here's your circuit up here, pick the gates to do that, I don't think many of you would have come up with this and said, oh, let me put together this piece of DNA, where these are the inputs A and B, and we hook this DNA together with these parts like this. I'm going to show you how this happens. But this effectively is the circuit, where if you have A and B, one turned on or off, as I say, and the colored arrows, for example, this brown arrow shuts off this arrow, this blue shuts this off, this green shuts this off. You could go through this and you'd logically see the only way I get yellow out is the truth table here when we have an XOR gate. So I said easy, huh? But it's not, and that's kind of what most of my work is how to create these using a software tool. So really quickly again, my group is called CIDR, which stands for the Cross-Disciplinary Integration of Design Automation Research. We make software. We make too much software. We can't maintain it. But when we can't maintain it, we give it to a nonprofit called the Nona Research Foundation. I started that because eventually my grad students don't want to answer emails about software they made 6 years ago. So this is a way for us to keep software in the community and make it available. And then as Pierluigi mentioned, from that open source ecosystem and my research lab, I've spun out 2 companies. One is called Lattice. It makes software to do what I'm describing. And Asimov uses software like this to make engineered biologics. So it makes engineered cell lines to make drugs. They're both in the Boston area. And then my software, I make a bunch of software. They all have cutesy names and icons. These are different software tools my group makes. And I'm going to focus on 2 of them today. Well, let me describe a little bit first. I'm going to focus mainly on something called Cello, but Cello and Eugene, they're high-level programming languages and design environments. You use those along with tools like Raven to design the DNA. So we describe what we want in these 2 high-level languages or environments, Cello and Eugene. They get planned to turn into DNA with something like Raven. Then they program robots with something called Puppeteer. And again, some of this will make more sense. So we have specified design build, then they go into bacteria. The tool like Clotho, which is the first tool I created, and I created this actually as a postdoc at UC Berkeley. That tool manages all the data. And what I'm also going to show is, and this is the hardware wetware co-design, hardware software wetware co-design, is we can, instead of making DNA, we can additionally make microfluidics that mimic artificial environments. And we actually get microfluidics out where we can do small scale experiments on these same living systems. I'm going to talk about Cello today, and I'm going to talk about Fluigi. I'm happy people that are in the audience are interested in this research, please reach out to me. I'm happy to point you at different tools or techniques or papers or answer questions, but I can only cover 2 today. So the way that most of this is funded, I'm going to get those 2 tools in a minute, is we've asked what kind of models of computation are available to biology, what communication patterns, what measures, what design rules, how to disseminate this, and we want to predict, scale, transform, archive, and learn. And so what this manifests itself is something called the Living Computing Project. It's an NSF Expeditions in Computing program. Some of you are familiar with that. And it basically explores 3 axes. It explains how we make digital, analog, memory, and communication devices in biology, how we measure them in terms of their space, time, complexity, and quality, and how we specify them, design, and verify them. And so there are people like myself, Wilson Wong, Mo Khalil at BU, Swapnil Bhatia, Chris Voigt, Tim Liu, Ron Weiss, Don Delvecchio. It's a little outdated. Swapnil in this picture now is at a company called Catalog DNA. But in general, these are the folks that are involved. And so Cello, one of the tools I'm going to talk to, is a digital specification tool. Cello stands for cell logic. And Fluigi is a design and verification framework for microfluidics. It also does specification, but it's focused on microfluidics. So Cello is the simple idea that you'll sit down at a computer and you'll literally will write Verilog. So we chose Verilog because it demonstrates the idea. We don't think Verilog is the best language for this, but it demonstrates you could use the same environment you use for synthesizing digital logic circuits and electronics, you could use it for biology. It goes into Cello, makes a circuit, and then becomes a piece of DNA. So it's this genetic synthesis tool or compiler. The first part of Cello is exactly what you'd expect in traditional logic synthesis. There's nothing special. It parses the net list, it goes through, and it makes basically makes a net list. Then what we have to do is we have to go through and manipulate that, because we typically, for the circuits we make, only have NORs and inverters. So again, you have to go through a logic transformation process to turn the net list you had into a final circuit that's only NORs and inverters, and then also minimizes it. That is all traditional EDA. Now this middle slide, this middle portion here now is we have our gate library. And if you see this circuit here, this is this circuit where I have to do a kind of a technology mapping step, where I have to map the gates that I have here to this circuit topology. And the first thing you have to realize is a simple coloring problem exists, that I can't use the same gate twice. Because the same gate in biology is going to consume the same, what's called transcription factor. It's still going to produce the same repressor. So they're effectively hooked together. If I choose two, in like in regular electronics, if I choose two gates, they're physically separated. In biology, it's separated on the DNA, but they are going to recruit the same molecules, the same proteins. They're going to turn on and off at the same time. So you have to have different, they're going to interfere with one another. You need to only use different colors. You can't use the same gate twice. So by definition, you can't make any circuit that has more gates than you have in your library. That's one limitation. But then you do this assignment, and then once there's assignment, you can see me. What happens is I can put a piece of DNA in one place and another piece of DNA here, or I could swap them. Because in my picture, I showed the DNA floating up and then shutting off a circuit. But when it floats up, it doesn't float forward or backward. It floats around in the cytoplasm of the cell. So it doesn't matter if one thing is before another, because there's no direction in DNA. And I'll show you an example of that. Think of a cell like a burrito. If you went to Chipotle and you said, "Don't let my guacamole touch my sour cream," they're like, "We can't make this burrito." That's like what my daughter tries to order. She doesn't want anything touching. You can't have that happening in a cell. Everything's touching. Whereas if I did that in electronics, I said, just mash all these layers together, your IC wouldn't work. Well, electronics has a very rigorous separation. Biology has the burrito separation. Let me go through this again. We have this NOR gate I showed you. What happens is we have this transfer function that I've shown where we increase the input protein and we get a different amount of output protein. That looks like an inverse relationship. The slope and the shape of these curves, all you have to understand is that these are all inverters and they all have different shapes. And that happens by how we tune elements of this DNA to turn on more strongly or more weakly, or to produce protein faster or slower. So that gives us different kind of transfer curves. And these shaded regions, what these show is for many of these systems, when these gates operate in that area, when you crank the input up too much, the cells die. They're overwhelmed. That's something that doesn't happen in electronics. I mean, obviously electronics fail, but they don't die. And so bacteria and living systems will die. So this gives another constraint on the problem, that if we want to operate in certain regions of these graphs, we can't actually do it. So again, I'm sure there's lots of questions, but here's the general composition problem. If I want to hook these two gates together in my library, here's the transfer curve for both of them. What happens is this orange one, I provided input. And if let's say I provide this level of input, and I get this level of output. This level of output then is the input here on the graph. So this level of output is now the input. So the y-axis on this graph then becomes the x-axis on the next graph. And so this is fine because the low output will nicely fit this curve and the high output will nicely hit this curve. So the low input hits a high output, the high input hits a low output. That's nice. If I just switch the order of these circuits, if I just say I want the purple one to drive the orange one, you'll see that this doesn't work. It can't actually hit the region of high on this orange gate. So this composition won't work. So it's the coloring problem I described along with matching those, what's called signal matching. You have to match the gates such that you get the behavior you want. And so you can see this little animation going. So part of the problem is we have this gate library. We can't use the same color twice, but you've got to keep doing this assignment to get it so that you get a valid output, so that all the gates can actually turn one another, each one on. We characterize them individually, and then we have to try to, and the goal here is to hope that composition works. And that's a big part of what this work shows. So we show that we can do this relatively quickly. It's like saying as we have more and more gates, how long before we come to a solution? We use different techniques. The most common one is a simulated annealing approach that ends up getting us to having a gate that a system that's functional. I don't want to get too much of the details of this because I won't finish, but the point being that these curves that I showed they have error bars, they have a thickness on them, because what they really are is for each of these levels of input, we measure the output, and the output is a histogram because every cell is slightly different. They have a range. And what happens when you are hook these together, you're able to have histograms predicting the output. So these are the inputs. This is supposed to be low. So this shows the general output of this cell. This is the general input. And what we're going to see is a good circuit. As you can see, there are some, the tail end of this histogram here starts to overlap with the beginning of this histogram. And so this is supposed to be a 0. This is supposed to be a 1. If you're not careful, this overlap could make them indistinguishable. So a good circuit has very good separation from the highest low and the lowest high. I keep losing my cursor. The highest low and the lowest high. The biggest separation there is in these curves, the better. That's a big part of how we score these circuits. So again, here's an example, very simple MUX, written in kind of a case statement in Verilog. This is a circuit for it. This is the genetic circuit at the bottom. And then what you saw, what we did is we took these circuits and we had predicted outputs. So here are the input combinations 0, 0, 0, 0, 0, 1. The black is the predicted distribution of output. The red is the predicted, the blue is the predicted, but then the black is what we got. And you can see we match pretty well. And so we did this for a bunch of different circuits. About 0.8 megabases. So a base is an A, a T, a C, a G. So about 0.8 megabases of DNA were constructed. That's a lot. We did this for almost all 3 input circuits, but we only published the results of about 65 or so. But at this point, we've done all 256. This is a little old at this point. It feels new, but it's relatively old. This came out in Science in 2016. So you can find the Science paper on this and for cello, and it describes this whole process. Some of them were wrong. We've since addressed why some of these mispredict. There's any number of reasons, but part of this is sometimes the DNA part of what this is that the recruitment of a protein in one location blocks proteins of others. There's physical binding of molecules to DNA. There's a variety of reasons some of these might not work, but part of this is the mind rules and the term of heuristics to make circuits that end up working long term. What's also interesting, if you think of some of these, is they actually implement other logic functions. And so it's kind of interesting that you might have a circuit that topologically looks like it should implement some function, but implements another. And so some of the circuits that implement other circuits might actually be a better implementation. So it's kind of this strange, you might end up using a circuit that for another logic function. And that's something that we could build into the tool. I'm not going to go through this. All this is saying is that we have, oh, maybe I will, I lied. This circuit has green, red, blue, etc. That's the coloring. But as I said, all of these lines of DNA, each one is the same circuit, just with the parts in different order on the actual linear piece of DNA. And you can see that the order matters, that some of them, this is the same circuit, but things order differently, you get a better performance. And so a programming language I made called Eugene, I also did this parts when I was a postdoc at Berkeley. This language is a way for us to say rules like before, after. We've evolved this to be like regular expressions, where you can write a regular expression and it will say, this is the order of the DNA, 0 or more of these, then this. And you can use kind of automata to describe how DNA is assembled. Again, Zoom is great because I can't see all the people with all the questions. I'm sure there are some, but we'll get to those. This is a quick example. I'm going to mute this. This is an old version of Cello, but you guys can see it's at cellocad.org. And the idea is right now what's going on is that the user is choosing how many inputs they want and how many outputs. Then they could write Verilog. The inputs are small molecular proteins, and the outputs are typically a fluorescent reporter, just as a proxy for an output protein. So you described your Verilog, you said how many inputs and outputs you wanted, you wrote it. It's running. Right now it's running that simulated annealing where it's placing all of the circuits, all the gates. So here there's an output that basically says, what did you try to do? This is that circuit assignment. And now you can see those transfer functions I showed. And it shows those transfer functions all lining up to produce a a certain score. It predicts what the outputs would be, the distributions for when signals are supposed to be low, what the output should be versus the high. And then it produces an actual linear layout of DNA for you. And it does this by using certain rules we know based on which part should be before which other parts for a variety of reasons, which ones might assemble, et cetera. And at the end of the day, this final piece is an actual DNA sequence that is that circuit. So if you actually sent this out for synthesis or made this, you'd get that functionality you described. So one of the things is I said, once you run out of circuits, once you run out of gates, you can only use one. Once you run out of colors, you can't do anything else. Something I was always interested in is, well, couldn't we take this circuit and chop it up, partition it, do almost some kind of cut, min cut, where you're trying to cut this into different sets. And then what you would do then is you would put those circuits in different cells. So you couldn't put one circuit into a single cell because it was too big. You didn't have enough parts, but you could put the circuit in different cells. And then they extrude proteins through their membrane. Certain proteins go through the membrane. That's actually how cells communicate to one another in environments, to tell each other about nutrient sources and do other things. You can use that same mechanism as kind of external wires. And now the cells aren't interfering with each other. But the problem is if you put two cells next to each other and say, talk to each other, they don't do what you say, they diffuse a protein. And so it's non-deterministic, it's relatively stochastic, it's unpredictable, it's got interesting effects for modeling, it's not a great engineering framework. And so I said, why don't we take those circuits, put them in something so they can't move, and connect them with channels, and then we could open and close those with valves, control them with a computer, and then we could also have these circuits, instead of making fluorescent proteins, we could make biological molecules that are detectable by electronics. They can produce free electrons, they can produce changes in pH, they can produce changes in temperature. We could actually monitor the system electronically and have that control the system. They could talk to one another, talk off of chip, and have these distributed networks. And so this is where I get in this hardware, software, biology co-design. So why not have Cello not only spit out a piece of DNA, but it's already got the graph of the circuit. Couldn't it spit out the net list, so to speak, for a microfluidic that could house it? And if it spit out the net list for the microfluidic to house it, couldn't we place and route that microfluidic and make it? So this is a figure many of you will be familiar with. When I show biology people, I point out that there's a part of hardware software co-design where what's hardware gets decided and what hardware is decided. We're going to leverage the same idea, but instead it's going to say this part of the circuit should be DNA. This other part should be the control software for the microfluidic. This other part should be the actual hardware for the microfluidic. So the wetware is the DNA, the software is the control for the microfluidics, and the hardware is the microfluidics themselves. And so someone would sit down at a computer and make their design. They would say something like, oh, I want this, and I want to also have some microfluidic. We'd get a microfluidic design out. We could make it. So something that I'm interested in is how we can make these very quickly and do prototypes. This doesn't help me if I have to make a microfluidic and it costs $1,000, you're going to be very leery to use my workflow. But if the microfluidic costs a couple dollars, then you'll make these quite quickly and do prototyping. And that's what we talk about, how to do it with mills. Then we can control it with computers. So we are making a whole bunch of tools to do this. Specify, design, build, and test. And I realized I've been talking for about a half an hour now. And what I want people to know right now is that one of the main tools we make is called Neptune. I'm going to show you that. But we've also, when I got in, let me back up. When I got into synthetic biology, I got into it because someone told me about a NOR gate. And then when I went to the biologist at UCSF to learn about it, I said, well, how are you designing this? And they go, oh, we're using Excel and Word. Like we're writing our DNA sequences in Excel. And I said, do you have a database? And they go, we know what they are, but we don't have any, there's no one that programs that works with us. The way I got involved early on was I wrote a database system for biologists at UCSF. And if you know anything about the group I was in, they were just, Alberto, the advisor, was like, just make sure that function is separate from the implementation, separation of these concerns. So I was like, well, we need to make a system that abstracts the biology away from the function, has parts in one place, and we map them in the middle. And so that's kind of how this all got started. And microfluidics are similar, where microfluidics as a discipline there's not a real standardization of net list format of how to fabricate it. They're nowhere near the level of sophistication of a semiconductor manufacturing operation. And so a lot of what I've built is I just wanted to make that microfluidic I showed a couple of slides ago. Just like I just wanted to make the bacteria talk with the NOR gates, but someone's like, we can't get there until we build the infrastructure. So a lot of what I'm going to talk about for the next 10 minutes or so is some of the infrastructure. And some of it is this upper left is a tool called Neptune, and some of it's a netlist format called Mint, Microfluidic Netlist. What I'm just going to do, a little motivating example, is that we've made a microfluidic that if you put in a sample that has blood and cells, well, sometimes you want to do, you want to actually do a test on bacteria. You need to separate blood from the bacteria. That's called acoustophoresis typically. And what you want to do is you want to have a sample go through 3 stages. You might go through many, but the first is you want to separate out the blood from the bacteria from the blood. This circle is, I'm going to show you a microfluidic that helps us do that. And this is a picture of one. It's a very simple device that what happens is you flow in this particular liquid, and then you flow in cells that have, you flow in another liquid that has blood and cells, and then because of particular vibrations of ultrasonic excitation, that heavy objects go one place and lighter objects go another. And because of the difference in size and density and whatnot of the actual blood and the bacteria, they go to different places. And so what happens is the cells go up or down where you can collect them, and the waste stays in the middle of the channel. And what happened with many people is they made this with glass, and it was expensive, and people modeled it, and the modeling was never quite right, and they kept trying to make mathematical models. And instead we said, stop making models, just make lots of them and try which one works. And they said, that's too expensive. And we said, what if we make it with plastic? And so we tried to make it, we made it with CNC milled plastic. And the way you can do this is that we have made a language for microfluidics called LFR, or liquid flow relation. It basically replicates a netlist. You don't need to understand everything I'm showing in the slide, but it's the idea that you have input flows and output flows. You can assign them together. So what I can do is describe a netlist that says I have some inputs, I have some outputs, I have a trap. I can start to describe the composition of a microfluidic. So like for the acoustophoresis device, it's very simple. We have an input flow. We have one location that's a separator that we're going to do with percent, and then we have 2 outputs. This goes into this tool called Fluigi. Funny, now I'm doing this with Luigi here. Fluigi with a play because somebody was making a Mario Brothers logo and they had Luigi in there. And then I was like, well, this is like fluids, Fluigi, and that name is stuck. So with my host in mind, this is a great name. So what's going on is somebody is basically able to write that liquid flow relation. So here they're writing the channels. They're saying, how should this be laid out? But they're just basically saying, I want one flow to go to another. And when this is compiled, it makes a netlist. So this is, again, a design environment for microfluidics. Because when I write this, I don't want to say the channel is this wide, or just like VLSI, I don't want to be specifying that. There are models underneath that actually take care of the geometries. You can just say, I want these flows to come together. And if you want to change the lower level pieces, that's what's being shown here, is you can go in and change the design rules. You can change the radiuses of ports, the size of ports. But again, you have this layer of abstraction. And again, this doesn't really exist typically for microfluidics still. I mean, people still design them in Illustrator or by hand. And so what's happening now is, again, it's running a tool and then it's making this microfluidic, so it ends up making the actual schematic for this. These microfluidics have 2 layers, a layer that controls them and a layer where liquids flow. It makes the 2 layers. The blue is where the liquids flow, the red are the valves that control it. So I couldn't go through this extremely slowly, but someone wrote that I basically want one input, that's on the left. I want to go through this serpentine mixer, and then I wanted to produce 2 outputs that are controlled by 2 valves. You wrote that at a very high level, then it placed and routed it. I mean, we still are working out bugs and kinks, like it's got this weird bump. Like, so some of my students actually are working with physical design people from EDA to make some of this software better. Makes a net list. I'm not going to go a lot into this because people aren't, the point being, we have a way now. We were one of the first groups, to my knowledge, that said we want an intermediate layer between this high-level language and the actual device that's called Mint, where we have flow layers and control layers, valves. We start to do this specification. We've talked about some of this work at ICCAD. So typically the way my life is now is I publish very little in conferences. It's primarily journals, but some of this work is stuff that could go into kind of traditional EDA conference venues, is how do we do in microfluidic design. Just that at the end, we have some of these layers are flow, some of them are control. We have different devices and they're all connected. Again, we have this net list. So the whole place and route pieces, we have to move everything around just like we would have to do in VLSI. With design rules that we have to respect for the sizes and geometries of the microfluidic. So moving this stuff around becomes a traditional CAD problem where we need to move, we need to move all the different layered objects around. Once this is done, you see that snaps to and actually looks kind of like what our microfluidic should be. For the Acoustophoresis device, it's very simple. Some people don't want to use that flow I showed you with Neptune. Some people want to do an actual CAD canvas. So we made a tool called 3DUF or 3D microfluidics. Because at one point we were 3D printing these and the name is just stuck. So this is a CAD framework where you can literally place the different layers. So if you go to 3duff.org, you can actually see this tool. You can play with it. It's been published in Nature Scientific Reports. What people do now is you can actually design your microfluidic. So it's better than Illustrator because Adobe Illustrator is more powerful, but Adobe Illustrator doesn't let you have the idea of selecting all the devices like I'm showing you here and sliding their sizes, changing all their parameters. This produces a file right now. It just produced a real file that we're going to be able to export in a minute, and I'm going to send to a mill. So this is basically just showing you sizing things, how you can place, connect, and then you export an SVG, a vector file. You can see it's the actual size of the device. It gives you two, not masks, but shows you the two layers of this microfluidic. And then what's going to happen is you can send them literally to a mill. So this is an off-the-shelf kind of small desktop mill. You drag and drop the design onto this mill, then it's going to actually mill this out of a thermoplastic. And the polycarbonate is one that we use a lot, and so it's actually going to mill this out. So again, we can actually mill these. Oops. Let's go to the next slide. We have a whole bunch of primitives. So again, I'm going to move on to the next topic, but that's kind of the flow, what we're doing. So the point being, I didn't want to build a CAD framework for microfluidics. I wanted Cello to spit out a microfluidic, and then I would just use existing CAD tools to make the microfluidic. But none of them existed because I needed a microfluidic design environment that was going to take a high-level net list from Cello. And nothing could do that. We had to design, and I was like, well, let me define the net list, and then I needed to make some design tools. So all of this journey has been to try to build this automated infrastructure to do some of these things. A lot of things to talk about, lots of pieces there. The thing I'm going to talk about here is the fact that what we have is this idea that we have an automated lab. So I make these microfluidics. I mean, we make the circuits in Cello, we make microfluidics, but where's the actual DNA get made? All this was in silico. Where does that get made? Well, traditionally it gets sent to a lab and somebody does it. You do all this high throughput design, and it sits in the lab for a couple of weeks, and someone messes it up. So you did all this work, and then it's down to the weakest link. So what we want to do is automate these. We want to have cloud lab. We want to automate this the same way we have foundries that automate it. We want automated labs in the cloud. This is an article saying that the lab that's automated isn't a bunch of robots that look like people. Like we're not going to have a robot butler in there doing something. We're going to have to automate and hook together existing lab infrastructure. BU and others are part of this global biofoundry consortium. We're trying to get different labs to talk about standards and round-tripping designs, kind of making such that if you just submit a design, it would get fabricated biology anywhere in this consortium. Just like when I send a job to Amazon, I don't actually know what Amazon Web Service actually processes it. It's like a cloud lab, but for biology. So that lab for me is called DAMP Lab. It stands for Design, Automation, Manufacturing, and Prototyping. We make microfluidics, we make biology there. This is an example of one of the work cells. What you see in the middle is robots on a track that move back and forth. And then they move DNA and other reagents and stuff around to other devices that carry out that protocol. When I go back to that very first slide I showed you, when I said, well, we have the circular piece of DNA and we cut it open and we put it in and we have to pick a colony and it grows and changes color. All of that process can be automated. It's just, you have to incentivize why to do it. And most academics, they're like, we'll just hire more grad students. And we'll just have them do more because they don't need to crank out thousands. But Cello could literally give you thousands of designs to make. And then what are you going to do? Just pick the best one because you're super smart? That's kind of how biology is on some level. It's still gated by some smart biologists having to figure stuff out. And biology now is cheap enough that doesn't need to be the case. Like the Moore's law of DNA design is that the ability to write DNA is getting cheaper and cheaper. And so we have more DNA than we know what to do with. So instead of just bottlenecking it and having someone figure out what's best, to me, it makes sense to build lots of things in a safe and responsible way and then screen them and learn rules from them. And that's what this work cell would do. Work cell does a lot of things. This chart basically says there's some that we can categorize them by different colors. They're in these different classes. We can do like 28 different kinds of things with this system. We could show how to do it with workflows. These are how you use it by stringing together symbols. This could be like an operating system where it's scheduled and batched and optimized and parallelized. So there's a whole operating system, computer science, scheduling piece of this that has nothing to do with biology again. So someone who's really hungry in this space is not well carved out by computer scientists, like how to automate labs. It's really important. And you guys all know, all of you, if I gave this talk a year ago, you'd all be like, why do we need to automate biology? Now, today, I think everyone understands why it's important for us to automate biology and biological discovery. So and I'm gonna get to that, This is a picture here of McDonald's. It's literally McDonald's. On the left here is literally the instruction manual for you've got buns and how you heat the surface and you go through and you assemble your hamburger. And then on the right here is for all of the sandwiches, what consumables they use. This is another, they have scheduled humans. We could do something similar for robots and protocols. The reason I use a McDonald's example is there will still be humans in the loop. So there's still a human scheduling problem here. McDonald's doesn't need to optimize, even though they're serving billions and billions of people, they're not making more hamburger. They're not making nearly the volume we would expect to make if we're successful in biology. Lab is a core facility. This slide is just showing that we offer services. We have a whole infrastructure. I'm cognizant of the fact that I've been talking for about 45 minutes. I'm gonna get to the end. We make, this is kind of a picture of the lab. We have robots and different hardware in the lab. These are costs of things. Okay. The last point, last 2 minutes or 3 minutes, or I don't know how many minutes, but it's gonna wrap up, is I tried to get that big piece of equipment for 3 years. And I wrote grants to NIH, NSF, and if I showed you the reviews, everyone's like, why do we need to automate biology? We can just have students do this. What's the robot gonna be used for? Why do we need protocols? Why do we need to program at high throughput? Why? And then COVID-19 hits. And clearly one of the bottlenecks is how we test, how we screen vaccines, how we do this. And before there had not only been something as urgent as COVID, but also something where timing is so important. Like, I don't think if you said, well, we'll just go through the process and we'll get a vaccine in 2 years, no big deal. I don't think people would be very happy. If we said, you can't go out of your house, you know, and you get tested in a year. Now, unfortunately, politics aside, we haven't done a good job in this country of scheduling testing, but even if we were doing really well, we wouldn't have the infrastructure to test 300 million people at the rate we need to. We wouldn't have coordinated that, and we definitely wouldn't have done it with humans, so we need robots. So this is a picture of my lab. That same lab I showed you Had one robot, COVID hit, university's like, oh, we better get you the rest of those robots you wanted. So basically from May to June, in the span of 1 month, BU committed millions of dollars to getting 8 of these robots up in the lab to run to do COVID testing through a testing that's called RT-qPCR. And the only way this is going to work is we need to process enough students. If we don't process enough students, the testing is kind of moot. We need to be able to test people regularly enough, which means we need to do about 5,000 tests a day at BU. But BU, what happens is we test everyone roughly once a week, maybe more, depending on their activity level. And this only happens because all of those robots and all that process is integrated. So that's what's happened here. And so it's a lab, it's got all of these kind of robots, 7 Hamilton stars, these other machines that may or may not mean something to the folks on this call. It's got a staff now of about 36 people. I don't run it. I prototyped it and set it up along with another professor named Kathy Clapperidge. Many people were involved. If this is being recorded, someone's watching YouTube, I'm not taking all the credit for all of this. Many people were involved, but my lab is set up for the robotics. And again, it can do about 6,000 tests a day. And then it's got some manual processes, but most of it is automated. So again, back to a lot of what I said, I couldn't sell this equipment to automate genetic circuits, but getting this, I couldn't sell this facility, but testing we could do. And you can kind of see the capacities. This is kind of the journey. I'm not going to go through all of this, but we intake samples. People self-swab at BU. You go to its testing station, you self-test. Then every day at regular intervals, these are collected. And then they go through a process where single tubes are put into 96 well plates. Then those 96 well plates eventually end up in these 384 well plates. Barcodes are tracked. The barcodes are separated from their identity for any number of confidentiality reasons, but there has to be a software system underlying all of this. That software system is called LIMS, or Laboratory Information Management. It does all sorts of tracking. For this audience, you might be like, well, this is in the weeds a little bit. Sure, but the only way this works is with computer scientists. And the only way this works is with scheduling software and automation and databases and tracking. So again, if you're a computer scientist, electrical engineer, and you're thinking, how could I have participated in COVID? You don't need to know biology because, I mean, that'd be great if you do, but there are lots of computer science problems that need to be solved just in automating this process. And the same, like, we're not building the circuits I talked about, but the same software for that circuit design could have been used for COVID. And I hate to be a downer, but I hope COVID is the last thing in my lifetime, but I suspect we're going to need biology for something else: materials, fuels, food. And I don't want my daughter, 50 years from now to say, oh, we're having a food shortage or something because we didn't get our act together with biology, or we're having some sort of energy crisis or global warming crisis that could have been addressed with smart fuels or something because people said it's too slow to do manually. That's something that I think, if I were to say what's a call to arms for this community, is to figure out a way to get involved with biology to help it become more efficient, more formalized, more of an engineering discipline. And if we do that, we'll be successful. Lots of testing. Long story short, BU has done almost, since July, has done almost 162,000 tests with that system, about 122 positive tests, which is a relatively low rate. You can see how it compares to the bottom to Massachusetts and Boston in general. We're doing pretty well. We have a 0.05% 7-day average positive rate. There's a whole process of quarantining and isolating students. And so the last, I'm wrapping up. The message here is, I can't sell this as a biofoundry for genetic circuits. I think that's cool. I can't sell this to people usually about applying computer science to biology, even though you guys all get this. I can't sell this about standards. I can't sell this as a biology service. All of those 4 points are exciting. That's why we should do this. It's computer science and biology, new standards. But the point is, none of that messaging got me my robots. A crisis got my robots. I need other people in this field to help me think about the real problems that we need to talk about, the mobilized governments, the mobilized universities, that say that computer scientists need to be part of this process. It can't be that, because none of us probably on this call that are computer science are probably going to cure cancer. We don't have the biological background, but we could automate the system of screening and testing and diagnostic that helps cure cancer if we could help articulate that and motivate it in the right way. So really, can the CPS community engage in this? Can you guys take what I threw out there, see where you guys might fit in, and maybe take it to the next level? So lots of people to thank. These are just a picture of some of the people involved in testing. A lot of the work has been going on for, almost 10 years now. I thank Pierluigi, I thank USC for having me come and chat. I'm looking forward to engaging with you guys. And with that, I guess, Pierluigi, I'll turn it over to you.
Wow, thank you very much, Doug. It was very inspiring, not only from the point of view of understanding this relationship within biology and design automation, but probably even understanding essentially the purpose of to give to research, and how actually sometimes the purpose is not, or let's say the result is not the one that we predict before, or we shoot to other dreams, right? But then our dreams come true when a crisis starts. And so I'm wondering actually if this was even one of the first levers that started also EDA in the first place, right? So It was hard to sell some of these concepts at the very beginning, but when the crisis arrived and the need for these processors and so on were the pull. All right, so for the research in this field. So I guess there are already lots of questions. So I some of them in the chat and some of them in the Q&A. So let's get started with some of them. I have also some, but I give priority to the audience. So actually already some feedback on the Q&A. Amazing talk. So can the idea of biodesign automation be extended to other abstraction levels? So you were talking about gate level, right? Yeah. And Shreya is asking about, what about system level or transistor level? Are there analogies with biological system and transistors, or more system level?
Yeah. I mean, there's definitely there's definitely multiple layers of abstraction. So I was saying like a NOR gate, and that being a DNA element. But then there's the A, T, C's, and G's. A's are already an abstraction of a chemical, of a basically chemical of adenine. So there are chemists that will work at a lower level, just like material scientists in a fab in a clean room will work at doping semiconductors. So there are people that might want to just tune the actual DNA sequence. There would be other people that might want to tune the device. People might want to tune the system. So the short answer is there's lots of different abstraction levels. It's harder to go higher for biology right now, just like it is with all things. If you keep abstracting, it doesn't really work in practice. But there's definitely different abstraction levels.
Yeah, and okay, so we have another question. Does the gate library size, so number of gate colors, depend on the kind of cell you're dealing with? If so, does that mean different cells can have different levels of DNA genetic manipulation achievable compared to others?
Yeah, so absolutely. So the gate libraries are specific for an organism, and they're probably specific for a certain strain. So we have a very specific strain of E. coli, and we may develop a gate for that. We recently published a paper that had a gate library for yeast, and that library is different. Bigger or smaller is not really the way to think about it, but it could be bigger, it could be smaller, it's different. If you have B. subtilis, that's another organism. Or if we had like CHO, that's Chinese hamster ovary cells from mammalian, or HEK, human embryonic kidney cells. There are different. The short answer is yes. The gates depend on the organism. So some cells might be better for certain things than others. So it's kind of like right now we're kind of spoiled because we're very familiar with like silicon. But it's not like you often make it, you say, should I make this in silicon or gallium arsenide? You're kind of working with one semiconductor, whereas imagine there are many more, what do we call hosts in biology? So the short answer is yes. So every time we run this, you'd have to run this in another organism.
All right, we got lots of questions on the chat, still about the number of gates and so on. So you emphasize that all the gates of the same type on a synthetic circuit have to be different though. So how many of the NOR gate equivalents then, for example, do you have for building a NOR gate? On top of this, I assume that there will also be connection constraints between gate. So here it's like, yeah, so how do you see the scaling of synthetic circuits in terms of compositionality? What is the largest circuit did you design using synthetic circuits?
Yes, good question. This has changed. So there is about 40 different gates. There are about 40 different gates. That number changes all the time. It's getting bigger and bigger, but that's kind of the size. We've made circuits that have about 20 gates in them. So from a brute force standpoint, like it's not like it's a simple EDA problem. If I just literally enumerated all of the circuits, then you're kind of done. So cello, as far as like a practical tool, is probably like, again, you just maybe even took a couple of days. You just enumerate all the circuits and be done, and just have a lookup table after that. So again, 20ish, 40ish number of gates, circuits that might have as many as 20. But that framework is in place as we get more and more gates, new organisms. Really what we end up using cello for is that what the earlier question was, as we get new gate libraries, we can just quickly plug them in and rerun them. So that's kind of the scale. We are working now on circuits that are intercellular. Again, those are circuits that are communicating. So we might have 20 cells, each with 20 circuits. So it starts to get bigger. But again, yeah, we typically probably won't, it's not really going to be about size long term. It's going to be about specificity of these gates, sense new things and produce new outputs.
So probably I'll also insert one quick questions. So I actually thank you for Fluigi, of course, but I was also very curious about the other system, right? The simulated anneal that you had, where you actually find either that what you thought it was a certain circuit, then it turns into another one, and so on. So this looks to me an example of emerging behaviors that probably can happen in biological systems, right? Where you think you can create a circuit by just composing things, but then you might get another functionality that is kind of emerging, probably. I'm not sure whether it can be seen like that. Certainly this phenomena is in biological processes. So I'm wondering what's your view in that, and whether you found a way to encapsulate this in the library beforehand, for instance, to predict this kind of phenomena that can emerge when you actually put things together that in isolation would look like and nor, and then probably together they come up something different.
Yeah, I think the closest thing, what we have done is, when it's kind of this builds on when we have this initial circuits and they don't function as we want, we're able to denote that and take a look at the structural composition of that and compare that to other circuits that work and start coming up with some rules. And we say, anytime we put this part next to this part, we get this strange behavior that we can't explain, but we'll add that as a constraint to either not do that or do that differently in the future. So we build those constraints. So that, to some extent, sometimes we can figure out, and we've built new pieces that prevent that from happening. So what I had these very basic parts. I showed something called the gene, something called the promoter. We have other devices. One's called a ribozyme. We have other small pieces of DNA that can end up isolating DNA differently, spacers that space the DNA differently to get different behaviors. So long story short is, we try to capture that knowledge. We try to create rules to prevent or bias toward it, but that's kind of the extent of where we've gone. Machine learning is a huge part of this that I have not really started to tackle. And that's another wide open area, is to learn from these circuits and use that knowledge for the future. I've done relatively little feeding this back into like biological knowledge. So someone could look at this as a biologist and say, oh, look at a new biological property I hadn't known. Let me add that to the canon of biological information. That's a loop that I have not closed really well.
I see. Great. So we have actually two more, let's say, questions that are more like on vision. And so one is actually on what is your main project recently? So what are you working on right now, in addition to the COVID that actually brought you to the front line, looks like?
Yeah. Oh, go ahead.
Yeah. The other is more like on visionary, another visionary question. I will ask this later.
Yeah. Well, we just published a paper. I mean, yeah. So I spent a lot of time now moving into microfluidics. So what we did, which not shown, is there's a paper. So many of you are familiar with temporal logic. We have a tool called Phoenix, which takes signal temporal logic and can compile that into circuits. Now, the problem is no biologist wants to write single temporal logic, like eventually, always, until, plus bounded, period. We did this with a professor named Colin Belta.
Yeah.
He's a control professor at BU. We have a tool that does that, but we're not going to keep moving higher. So after that, we're done. We don't need to keep moving up the abstraction. So one thing is this temporal logic compiler. The other is I want to have the biology communicate with electronics. We need to embed electronics on the chip, and our chips need wires, and we need to manufacture them cheaply. So a paper we just put out in lab on a chip is how to use ink that is conductive. We can wipe ink on the microfluidic so they can create wires, because we don't have a way to deposit metals. Because we can do lithography with some chips, but most of the chips are not. We don't work at that because it's too expensive. It ends up defeating the purpose of these rapid prototyping devices. So we're trying to integrate electronics into chips, custom sensors. NSF has a program called SemiSymbio, which is the Semiconductor Research Corporation sponsors, SRC, and they have a thing called SemiSymbio. NSF just had a call for that. We were one of the people awarded that, and that's how to integrate electronics and microfluidics and memory. We have biological memory. So imagine that I had a bacteria and I could ingest it, and it would go through my gut, and every time it sees an event, it would flip a bit. And at the end, I could harvest that bacteria and read its DNA and look at all the times the bits flip in different places. And that it would encode, I saw, let's say 8 bits, you'd have 2 to the 8 different states it could have seen. You can literally get DNA to flip itself. There's something called a recombinase that flips DNA. So the point is we're doing work on integrating electronics, work on higher level languages, and work on integrating memory into all of this. I didn't talk about sequential logic circuits either. I mean, we've made some SR latches in bacteria as well. And again, that's not to make memory like gigabytes of memory, but that's because these biological systems need to have a memory. They need to remember what they've seen.
Yeah, actually, yeah. Interestingly enough, right today in our CPS design course, we were having a discussion session and some paper also touching upon signal temporal logic, and I exactly got this question. So can be actually used? It was used in a just CPS classical control system, let's say not live system, but indeed. So they were asking me, can be generalized, can be used in other contexts and so on. And so now for those that are attending, I think they got also some more.
And for those of you who are grad students, the reason that paper's not out is the student graduated. And he promised me he'd finish it. He's still working on it. We're going to finish it. But for all you grad students that graduate, you're going to have the best intentions of finishing papers. My suggestion is finish them while you're in school. Don't wait till you get out.
Right. Yeah. Good. Good. Lots of good suggestions today. So something more on actually the concept of collecting biopsy from rats' brain and utilize this in a circuit for processing data and also memory at the same time. What do you think about now tampering with the brain? I think it starts to be the question here. And when that can be realized as application in industrial revolution. Also, can we feed these cells by only electricity and air as a source of energy to have them survive?
So the latter question about how cells live, both of these questions are a little bit outside. It's a good question. It's a little bit outside my area of expertise. How cells survive, there are extremophiles where we find cells living in like extremely hot, like by volcanoes, right? And things like that. I'm sure there are some species that are very robust and maybe feed out. I mean, there's all kinds of bacteria that can do things. We can't feed lab strained E. coli just air and electricity and have them survive. They need to be plated on some media that they're consuming, or they need to be in something called a chemostat that's continually keeping them alive. So the short answer is maybe there are some that you can have only electricity or air, but they'll be extremophiles. I don't know what those would be. And then the idea of getting a biopsy from a rat's brain and utilizing it in a circuit. That sounds like a proposal for NIH. So the short answer, so the real issue, what you really want with biology, you could maybe, let's say you could do that. It's kind of like if I said, can you take, and I picked some strangely adjacent semiconductor on the periodic table and said, could you get a inverter out of that? You could, but you wouldn't, but you can't, you want to be sticking kind of, you want to, right now, we don't want to really be expanding the biological things we're testing. Like we'd be happy if we could just get this to work regularly in E. coli. Like part of, like when we go to a fab, we don't have people making BJTs and CMOS and PMOS. There's one transistor technology, and they try to get that to work really well. Now there's reasons to have other ones, but we're trying to, at least my group is very focused on picking one technology, which is kind of transcriptional circuits in bacteria. So I don't know if that answers your question, but maybe you should write, it sounds like you could write the next grant, something about rats and biopsies. I don't know.
All right. Well, I think we are well beyond the allocated time.
Okay.
And yeah, I think, yeah, all the question, like if there are other, oh yeah, so that's actually, there is a thank you. Yeah, more feedback on the, if there are other questions, you're still welcome. And by the way, I mean, you can also raise your hand and ask the question in person if you wish, not only on chat. Yeah, so.
I'm happy for most of you. I mean, I think, again, Doug Densmore, I'm not hard to Google. Feel free to, if you have questions or something that you think about later in the day, feel free to email me. Just put USC seminar or something in the title of your, so I'll know that, because I'd love to say I answer every email, but if I know it's from this seminar, I'll get to it. The other thing is, I don't know who's on this call, but if people are, you know, I'm not trying to poach people, but if there's anyone looking to do postdocs or any undergrads looking for grad school on this, and you're interested in this research, please reach out. I'm actually hiring postdocs now. So yeah, spread the word or let people know. I'd love to have some good candidates come. It's a little colder in Boston than it is USC, but it's still a fun place. And Boston is actually, if you're interested in biology, Boston's a great area to be. So.
Okay.
Yep.
Thank you very much, Doug. So,
all right, thanks everyone.
Yeah, see you everyone next Wednesday, and thanks again. So let's thank, we cannot make an applause, but yeah, virtually we will. Thank you, Doug. See you.
Okay, I'll see you later. I'll be leaving too.
Bye.
Okay, bye-bye.